and together with like-minded people like you, me, and other people who would be joining soon or in near future, definitely we would be taking this Scholar Lab to greater heights.
Thank you, sir. Pleasures
Now,
are all.
coming to this opportunity where I can connect with other people, let me share the screen first. Uh, if if I have, yeah, I am able to. I'm sharing the complete screen now. I hope my screen is visible.
Yes, sir. It's visible.
Yeah, and this is where I share the presentation. I hope now the complete presentation is visible to everyone.
Yes, we can see the first
Yeah.
page of the presentation, sir.
So the title of the presentation, today presentation with Scholar Collab, is Nanoparticles Advancement in Skin Surface Therapy. Coming to this nanotechnology, like nanotechnology is one of the very new branch of science, although it has been developed in the last decade.
I think there is some electricity cut onto professor side. Meanwhile, I would request to our joining over here. It's a very little request. I would be sharing a form with you all into this chat. This form will be used into generating of your certificate in collaboration with IEEE MTT-S. So it's my humble request from all of you to take out your couple of seconds to fill out that form so that it would be smoother to generate the certificate for you. Thank you.
Am I audible, guys?
Yes, sir.
Yes, sir.
Yeah, I think there was sudden drip in connection, so I think I got disconnected. Let me share my screen first again. I hope the screen is visible now to everyone.
Yes, sir.
And yeah,
Yes.
let me share the presentation. Yeah, so coming to the presentation, the topic of just give me the topic of presentation is nanoparticles advancement therapy. What I was discussing was that nanomaterials have been, it has been almost a decade where I would say, just give me a second. There is one urgent call over here. Yeah. So nanotechnology, it has been a decade since nanotechnology has come into the picture. And the way nanotechnology has been developing, we are seeing a lot of advancement, not only in skin therapy, but there are drug development, mechanistic development, coatings, so many different things. And the way nanotechnology has been developing, we are seeing the effect of nanotechnology into space as well. The rocket launchers, the rockets which are basically going into space, they are being coated using nanotechnological development with materials which are anti-radiative. So nanotechnology has immense applications, and we haven't used those applications even to 1% as of now. You may be aware that especially in cancer therapy, a lot of nanotechnology based medications have been utilized, and it is coming into the picture. So lipid based nanoparticles have been approved to be used as a drug carrying vehicle, especially in cancer biology. Coming to this detail, why I am more interested into skin therapy, because in this particular section in nanotechnology, I basically worked on serum oxide nanomaterials. Now coming to this serum oxide nanomaterials, it's our next generation therapeutics material, especially for diagnosis of drug delivery applications and other aspects. So if you go through these kind of material, what you will find is that this serum oxide nanomaterial has applications in anti-cancerous activity. It also acts as a cytotoxic material. It acts as an antibacterial material. It has been found to have activity or applications in neurotoxicity, genotoxicity, and more specifically, this serum oxide nanomaterial has been found to have a very high antioxidative activity. So this particular serum oxide nanomaterial, it finds its applications in multiple different sectors. Now, coming to nanotechnology, what exactly is nano? People do believe that synthesizing a material at its nanoscale range, that is where they achieve nanotechnology. No, to develop a material it's such a small scale range so that it can have its multiple applications. So nanotechnology is helping considerably to improve, revolutionize many technology industry sectors: information, homeland security, medicine, transportations. So there is no field which has left unanswered by nanotechnology, whether it is your smart infrastructure, whether it's your digital buildings, machine learning, soil and water, artificial intelligence, design, energy, anything has not been left alone by nanotechnology. This is why nanotechnology is finding is much and more application in all the different sector. And as of now, in this today's world, waste management is one of the area, water management is one of the area, transport is one of the area where lot and immense applications of nanotechnology is coming around. And we are trying to develop so many different types of nanomaterials based on their coming applications. So for those who have been not aware of this nanotechnology area, what you really need to understand is the size of biomolecules. We hear about blood, DNA, virus, and all these different kind of molecules. What are exactly the size of those molecules? Let's suppose we have a glucose molecule, the basic energy source, which our body which we take. Protein, DNA, virus, these belongs to those kind of size range where 1 to 2 nanometer, 5 nanometer, 10 nanometer, in those size range we can synthesize many nanomaterials, especially in those category range: micelles, liposomes, dendrimers, gold nanoshells, quantum dots. We can synthesize polymers in those nanometer range, and those nanometer range basically comes under nanotechnology dimension. Like anything beyond 1 to 100 nanometers comes under nanotechnology dimension. But in nanotechnology, there are two major points which basically help and which makes nanotechnology a very exciting. The number one, nanoparticles are similar in size range to many common biomolecules. When I say many common biomolecules, it means I'm talking about glucose, I'm talking about proteins, I'm talking about DNA, I'm talking about bacteria, those common biomolecule size ranges. So if you are able to synthesize, if you are able to fabricate any kind of material in that size range, yes, you are challenging nanotechnology. And the second important point is one can easily control and tailor the properties of nanomaterials at nanoscale range. Now you need a cloth. What you do, you go to a tailor giving a broad piece of cloth. What he does, he cuts that nanomaterial as per your size demands. This is where in nanotechnology what we do, we cut those nanomaterials in such a way that they can be synthesized as per the need, and as per the need your application depends. So these are the two important features of nanotechnology, and especially in biology, which basically helps in the development of nanotechnology area. Coming to serum oxide nanomaterial, now this serum oxide nanomaterials are very high antioxidative nanomaterial ranging. They are free radical scavenging and are of regenerative nature. For example, the serum oxide nanomaterial basically it exists in two different forms. For example, you see this serum oxide nanomaterial exists in serum oxide plus 3 oxidative states, and then again this serum oxide nanomaterial exists in plus 4 oxidative state. The moment it exists in plus 3 oxidative states, the serum oxide nanomaterial behaves as a superoxide dismutase mimetic activity. Superoxide dismutase means it scavenges superoxide radical. And when it is in present in 3 plus 4 oxidative states, it behaves as a catalase mimetic activity. And in catalase mimetic activity, what it does, it behaves as a hydrogen peroxide reducing agent. So in both ways, what it is doing is it is reducing your oxidative radical, and that is where we say that serum oxide nanomaterial is of very high oxidative, very high antioxidative molecule, and it depletes that oxidative stress molecule. Now coming to the application of serum oxide, or coming to the application of reactive oxygen species, how does the reactive oxygen species works? So reactive oxygen species, I say that these reactive oxygen species are inherent part of our metabolic system. So any kind of metabolic reactions which takes place in your body, you have reactive oxygen molecule coming around. You have reactive oxygen molecules coming around, and they damage those oxidative molecules. For example, if you have reactive oxygen, high level of reactive oxygen species, it damages the DNA molecule during oxidation of proteins. If you have a high level of reactive oxygen species, it leads to MMP activation, which particularly leads to metastasis, which is a part of a cancer. You have pro-inflammatory cytokinin release if you have high level of ROS. There are chemokines, cell differentiation, cytokines in presence of high angiogenesis. Growth factors are basically released in presence of high level of reactive species, and apoptosis, autophagy, altered cell cycle. So all these kind of different unwanted metabolic components takes place in the presence of high level of reactive oxygen species, and that is why balancing these reactive oxygen species is very much necessary. So that is why it has been said that involvement of ROS in biological process suggests the role of endogenous and exogenous ROS regulating cellular process. Now, coming to how this ROS basically get involved in the body, ROS are highly reactive and quickly translation between species. So what you can say that we as a human, whatever amount of oxygen molecules which we take, it gets it gets converted into superoxide radical. And you can see here is that you have superoxide radical, you have peroxynitrite radicals, and there are other different acidic conditions present in your body. You have superoxide radical getting converted into hypochlorous acid, and you have a hydroxyl radical, which is one of the fastest reacting molecule which is present in your body. You have a hydrogen peroxide molecule which basically depletes from your passes through the cellular membrane, and which gets released into the cellular system as well, where it is being functionalized by catalase and glutathione as peroxidase, where it basically goes to degradation of this molecule. So in your metabolic system, lot of oxidative stress are being produced by different molecules, and these molecules are scavenged, or it's needed to be scavenged by the different kind of antioxidant molecule, and this is where serum oxide molecule works. Now coming to the different diseases which are part of this oxidative stressor which happens in your body. For example, if you have high amount of superoxide radical, you will have mitochondrial enzyme, peroxidase enzyme, and pesticides. They basically lead to high amount of superoxide anion. They further leads to successive formation of peroxynitrite, and lipid nitration and oxidation takes place. Protein damage takes place, and we discussed DNA damage and protein damage, lipid peroxidation. So what is ultimately what we need to do, or what the body needs to do, is to scavenge these oxidative radicals which are produced in our body. You have seen how a human body ages. What happens in a human body ages? You can see when you are at this stage, your oxidative level or antioxidative level of your body would be entirely different compared to 10 years down the lane. The 10 years down the lane, the role of antioxidative metabolism in your body would be much lower, and that is why you see nowadays even doctors and so many health professionals are prescribing to take more and more antioxidant rich food, antioxidant based medication in order to keep your cells healthy. ROS generation and degradation, as we initially discussed, that ROS generations and degradation is an inevitable part of your metabolic reactions, and it is very necessary to have a very good level of antioxidative balance in your body compared to the kind of oxidative molecules which are present in your body. And there are so many different metabolic pathways which are involved, so many different enzymes are involved into that particular category. Now how these damaging happens in between body? So we have damaging controlling of reactive nitrogen species. So if you have an oxygen molecule, which basically we as a human we utilize in mitochondria to generate energy, the oxygen molecules, and if these oxygen molecules goes into mitochondria for generate energy, it gets converted into superoxide radical or peroxynitrate radical, and successively this peroxynitrate radical and superoxide radicals get further converted into hydrogen peroxide radical. So if you have any system which is able to scavenge this superoxide radical, or any metabolic system or any molecule present in your body which is able to scavenge this hydrogen peroxide radical in your body, you would be able to produce less number of oxidative stress, less number of oxidative stress inducing molecule, including peroxynitrate and hydroxyl radical. Now coming to hydroxyl radical, it is one of the highest reactive oxygen species molecule which can react instantaneously, and it can dismutate any kind of protein. And this is where your serum oxide molecules plays a role, because serum 3 plus ion basically it dismutates superoxide radical, and serum 4 plus ion it basically dismutates hydrogen peroxide radical. So based on these two roles, these serum oxide molecules come into the picture. Now, understanding serum molecule. So serum is a molecular weight of 58. It's a very rare earth element present of lanthanide series, and you can understand lanthanide series. You have the electronic configuration of 4f2 6s2 orbital. What happens in that is you have 4 electrons which are present in the final orbital, and this switches in between the different component. The molecular atomic weight of the cerium is around 140.11, atomic number 58. The crystal structure, develops an FCC based crystal structure. Now, what are the three different unique regions, unique nature of this serum oxide nanomaterials that what we develop as a serum oxide nanomaterials? The number one is redox nature, switching of oxidation state between cerium plus 3 and cerium plus 4. So you can see here how this serum oxide nanomaterial it switches from cerium 3 ions to cerium 4 ions. The second unique nature of this CNP is oxygen defect. So when you have synthesized a serum oxide nanomaterial, it creates oxygen defect. Oxygen defects are basically, you can see these hotspots. These are holes which are filled by oxygen molecule, and this is where it can easily adsorb those oxygen molecule, and these are called as hotspot for catalytic activity. And the third is potential antioxidants. The moment it is generated in form of a nanomaterial, this molecule behaves as superoxide dismutase mimetic. Mimetic means this in human body. To let you know that in human body we have two antioxidative enzymes which are present, that is superoxide dismutase and catalase. Now the best part of this serum oxide nanomaterial is that this serum oxide nanomaterials behave as superoxide dismutase mimetic and catalase mimetic, and that is why it is a very high antioxidative molecule which is present in our body. Now, this is the model of a reaction mechanism which indicates how a cerium 3 plus ion basically gets converted into cerium 4 plus ion. What you can see here in one reaction, this is cerium plus 3, where it reacts with hydrogen peroxide, it gets converted into cerium 3. That is, you can see how this hydrogen peroxide molecule is being utilized. You see, you have cerium 4, and you have cerium 3 in a particular structure, and then there is a release of H2 plus ions. Then again, the cerium 4 gets converted into cerium 3, and then again the release of oxygen, and this serum 3 plus ion is converted. And again, how you see how the serum 3 plus reacts with H2O2 and then gets again converted into serum 3 and serum 4 H plus ion. So this is a particular reaction which keeps on undergoing in a metabolic system, and you have your hydrogen peroxide molecule, you have your superoxide radical molecules keep on being utilizing in this particular system, and that's how this complete regenerative nature cycle of serum oxide molecule, you have a complete antioxidative system which degrades the oxidative stress. Now coming to this particular system, where I'll be discussing some of those papers which we have worked on years back and which we have published, and those papers are pioneers in this particular field. So there was a very initial paper, although I was not part of this particular paper, but it was published by our group back in USA, and Dr. Seal and Craig Neal, who were leading this particular paper, they detected picomolar detection of hydrogen peroxide molecule, uh, using serum oxide nanoparticles. So they developed a particular methodology where they were able to detect hydrogen peroxide in blood. As to let you know, even as a human blood, human system, we have hydrogen peroxide which is present in our body, and peroxidase, which including hydrogen peroxide as a part of reactive oxygen species present in the body. So we really need a particular system in order to detect what is the level of hydrogen peroxide in your blood, so that you can understand what is the level of oxidative stress, what is the level of antioxidative defense which is present in your body, and this is where you have H2O2 working as a particular system. So we were able to detect a picomolar level of hydrogen peroxide. We were able to detect even zero point 0.1 picomolar of hydrogen peroxide with a range of 1 micromolar, and this particular range was able to detect. The sensor which was developed was able to detect in this particular range. Now this picomolar detection was so accurate that if you change the system, if you change a molecule, the system was able to recognize, like, when we do a particular sensing development, we need to understand that is it possible to have this molecule, like, if it can be can any change in alternation of any kind of molecule, is your system is stable to understand that. So what you can see here is that you see this red mark, you have red mark is uric acid, sodium nitrate, glucose. So anything addition of sodium nitrate, glucose, you see there is no change in the sensor. But the moment you add hydrogen peroxide, you see there is a shift in the current in this particular system, which indicates that the moment you have hydrogen peroxide in your system, the sensor is able to identify that. And this is where this paper was published in scientific report back in 2017. So with this, we moved on to the next level of serum oxide nanomaterials, and we tried to develop those serum oxide nanomaterials in variety of different sector. And this is where you can see that this serum oxide, this was another paper which we published back in artificial cells, nanomedicine and biotechnology 2018, where we developed a serum oxide nanomaterials at the nanobio interface, a size dependent cellular uptake. So what you can see here is we have variety of nanomaterials. We have serum oxide nanomaterial at 10 nanometer range, we have serum oxide nanomaterial at 20 nanometer range, we have serum oxide nanomaterial at square shaped, and we have serum oxide nanomaterials in terms of rod shapes as well. So you can see that same one material can be synthesized into so many different aspects, and the size of this nanomaterial vary. We have in nanometer range, we have vary in 3 to 5 nanometer, we have nanosphere vary 15 to 20 nanometer, nanocubes of 30 nanometer, nanorods of 50 to 200 nanometer, and these are all the different nanomaterials. Now what happens with this nanomaterial is that the moment you give these nanomaterial to a particular cell, the cell needs to uptake those nanomaterial. Now this is where the serum oxide nanomaterial is not uptaken by this particular cell. It doesn't take serum oxide nanomaterial in terms of rods, it doesn't uptake serum oxide nanomaterials which is present in form of a cube, neither in form of spheres. What it really takes is nanomaterials in form of ultra low size range, and that nanomaterials easily enter into the cellular system. And what we believe is that the cells have a passive mode of uptake where it doesn't utilize any kind of energy, and it uptakes those nanomaterial. And this is where we say that low size range of serum oxide and nanosphere could efficiently be used for drug delivery application. And this is where our team tries to work onto those kind of nanomaterial to uptake those nanomaterial for any drug delivery applications. What you can see here is there are energy dependent pathways, then you can see that even if you have energy dependent pathways, the cell is not able to take any kind of material. So what we have done is we have used drugs, and energy dependent pathways have been blocked. Now the only mechanism which can work in this particular pathway is energy independent, means passive pathway. And in presence of this passive pathway, what we can see here is that serum oxide nanoparticle is being only uptaken by those cellular system. We also try to develop these serum oxide nanomaterials to prove that this serum oxide nanomaterial works in dual functionality. What you can see here is variety of serum oxide nanomaterials which have been synthesized. You can see this is small size range of nanomaterials. Now this is catalase mimetic activity, and as you can see here, as we increase the solution concentration, the catalase mimetic activity is increased, and this is sodium mimetic activity. And the moment we increase cellular system, the sodium mimetic activity is increased, and these all materials are completely non-toxic to a particular cellular system. Now in a similar way, we engineered these nanomaterials and to see effect if can this serum oxide nanomaterial have any effect onto the ultraviolet B radiation effect. And this is where we found that the engineered nanomaterial, Anvika, please, the engineered nanomaterials modulate neutrophil oxidative response to low doses of ultraviolet B radiation through the inhibition of reactive oxygen species function. So what you can see here is that you have ultraviolet B radiations in presence of serum oxide nanomaterial, and what it does is under the low doses of UVB radiation, the neutrophils activations are taking place, and that collaborates for photo damage of surrounding cellular system. But however, in presence of serum oxide nanomaterial, the NADPH oxidase and other different components comes up, and you have low level of ROS and high level of catalase and SOD activity which is present in the system due to presence of serum oxide nanomaterial 1 and 2. And this is where the effect of these nanomaterial 1 and 2 shows that even in presence of ultraviolet B radiations, low doses of ultraviolet B radiation, especially for those countries where we lie in tropical region, whether it is Brazil. This research was done in collaboration with Brazil, India, China, and other those countries which close to the tropical region. These kind of ultraviolet B doses radiations is very important, and they have lot of negative effects onto that. Now, coming to exploring the different role of serum oxide nanomaterials, we use this particular serum oxide nanomaterials in conjugation with miRNA for efficient diabetic wound healing. As we all know that in diabetes is one of the leading disease in the world in all of the major countries, and especially in case of diabetes, the wound healing doesn't takes place efficiently. So what we use is that in case of diabetic wound healing, we use serum oxide nanomaterial conjugated with miRNA molecule to see the effect of these antioxidative nanomaterial, and also to see the effect of these CNP nanomaterials in delivering the miRNA directly onto the wound healing. Now, to all those people who doesn't understand wound healing, let me tell you that wound healing is a 4 different stage, where in the very first stage you see you have a blood wound healing aspect taking place, and soon once the wound healing starts, you have an inflammation zone where fibroblasts and macrophages are part of that particular system. And once this inflammations and everything comes in, you have a fibroblast proliferating, that is proliferation of fibroblast takes place, and slowly this fibroblast proliferation leads into remodeling of the tissue, freshly healed, and epidermis takes place in case of this one. But if you have a diabetic condition, these particular inflammation, proliferation, and remodeling phase gets much more delayed, and this is where your serum oxide nanomaterial with miRNA basically helps in. I'll show you how this particular takes place. So you can look into these particular data, which are a mouse data. You can see how this PBS, you can see that what a different kind of nanomaterial, this 110 to 100 to 1000 nanogram of material, how this efficient wound healing takes place. And the normal wound healing, which basically takes place at the 8 of days 18, is presence of this 1000 nanogram of molecule. It gives days into in terms of 18. Now, this is the representative digital photos of a diabetic pig bones treated with PBS compared to 1300 nanogram of CNP miRNA, and you can see how this particular wound in this category is healing faster compared to the other normal wound. And that's how serum oxide nanomaterial, the role of serum oxide nanomaterial in delivering this particular aspect takes place. Can we grab this? Leave this. So we have this particular a very lucrative serum oxide nanomaterial conjugated with microRNA 146A for correction of impaired diabetic wound healing in diabetic mice, and this result is very promising. And very soon you will see this particular, just to let you know that this CNP miRNA is under clinical trial as of now, and maybe another 5 years to 10 years down the lane, you may see that this particular drug has been approved for diabetic wound healing, because this is one of the major case is coming around is diabetic wound healing, especially in this cases. So these are certain other data which show that how the diabetic wound compared to non-diabetic wound heals better under when you have CNP miRNA molecule, how your collagenous area gets much more better developed compared to other different areas in prescription. Another sector where we have used this CNP miRNA 146A is improvement of lung injury. So as you know that during the age of COVID, when COVID was going on, lot of people were suffering from different lung related complications, and what we found that you also need to deliver certain kind of molecules using lung using nanomaterial, and this is where we used our CNP miRNA 146A, and we found that lung functions improves with delayed treatment with CNP mRNA following acute lung injury. And these are certain data which are here, where you can see that you have controlled bleomycin and bleomycin with CNP mRNA 146A at day 3, and then you have bleomycin at CNP mRNA 157 at day 7. And what you find is that at day 3 and day 7, you have much more better recovery compared to other different kind of molecules. Now we kept on utilizing this serum oxide nanomaterial for other different aspects, and this was one of the area where we were trying to develop certain smart patch for visible sensing of oxidative stress. Now you all know that oxidative stress develops inside our body. Don't you think that it would be really fascinating to see if my wound is healing with respect to that particular bandage? And this is where the idea works in, where we developed a particularly seal patch infused with amplex red, which was able to get into your wound and which was able to indicate that yes, your wound is healing with respect to this particular patch. Now this is a data which animal model data, which you can see that a wound was created into a mice model and the patch was applied. Now you see here is that you have all these patches, and then suddenly at the end of 24 hours one patch is turned into completely green. Now this patch indicates that this wound has a lot of oxidative stress coming into this picture, and by looking at this particular patch you can say that your patch is turned pink because of high number of oxidative stress. It means you have to take this patch out and you have to utilize another some drugs for antioxidative therapy compared to your normal uh, patch. So this indicates that you are able to visualize the wound healing process from time to time, rather than like you do not have any kind of marker or any kind of analysis. So these kind of paths are highly helpful in understanding if your wound is healing properly or not. And this is an in vivo animal model studies based on silk based visual oxidative stress sensing patch. In a similar way, once we explored the patch, we also went ahead to explore any kind of hydrogel. If we can develop a hydrogel with sustained release of microRNA of serum oxide nanoparticles to promote wound healing, and in this direction we were able to develop one hydrogel which was working good. So these are the photographs of showing the deuteranic cryogel. So basically you can see how this particular hydrogel has stretching feasibility, compression release, and self-healing property. And this particular hydrogel is lot of property where over the course of time it was releasing slow release of CNP miRNA into a mouse model, and it was helping us into developing uh, a delivery vehicle for this particular CNPRNA. And not only into this CNPRNA, we also developed enzyme-free plasmonic biosensor for direct detection of neurotransmitter dopamine from whole blood cell. So this particular serum oxide nanomaterial, it was so accurate and it was so reactive with certain specific component that they have been utilized into a particular kind of a sensor. What we see here is you can use the serum oxide nanomaterial, the C4 plus. What you can see here is C4 plus 280 nanometer, and then you have serum 3 plus at 253 nanometer. They were so specific in reacting with dopaminergic molecule, especially with dopamine molecule, that they were used in development of a particular plasmonic biosensor. And we developed this enzyme free plasmonic biosensor for direct detection of neurotransmitter dopamine from whole blood cell. Now coming to this serum oxide nanomaterial molecule, we also mixed these serum oxide nanomaterial molecule with some other different kind of molecule, basically nitric oxide donor molecule, to see how this molecule act as an antibacterial. Because you need to have a molecule which is multifunctional, and this is where your serum oxide molecule was found to be highly multifunctional. So when we mix this serum oxide nanomaterial molecule with nitric oxide donor molecule, what we found that it was not only antibacterial but it was antifungal as well. And this property of serum oxide nanomaterial was highly appreciated. So what we found here is that what you can see here in this ppt is that not only E. coli but it was also inhibiting other kind of a fungal species, that is Candida albicans. Once the moment you increase the CNP concentration to 3.5 millimolar, you can see it is inhibiting only not only bacterial concentration which was inhibiting, but it was also inhibiting Candida albicans species and other at different higher concentrations. So together with this CNP and nitric oxide donor molecule, it was working like a charm to degrade bacteria as well as fungal species, working as a complete antimicrobial material. Now coming to my major research interest, which is also in towards sustainability directions, I do work in that category where we develop nanotechnology based solution towards sustainability, applications of nanotechnology for the development and improvement of biotechnological process and product, where we use eco-friendly, cost-effective, sustainable and safe materials. and especially in this particular direction, green and sustainable nano product technology materials are priority. So what we basically do is green synthesis. Now the reason behind choosing this particular stuff is that world is moving more and more toward pollution. More and more pollution is coming around, and we are making the environment toxic. So when you have a green synthesis of nanomaterial, you do lots and lots of waste prevention activity, you do economic activity, you synthesize less hazardous systems, safe chemical design, you have energy efficiency, renewable feedstock, you have reduced use of derivatives and design for degradation. So these kind of research, it really needs to be uptaken. Why? Because this is where the future of our environment, our planet is going on. We really need to be more onto green cities, and that is why one of the major research arena of my lab as of now is nanotechnology based solution towards sustainability. And in this particular direction, we have done lot and lot of serum oxide synthesis using green materials. For example, ammonium hydroxide and high temperature calcination treatment based serum oxide preparations we have done using serum oxide nanomaterials. We have done using cesium cumene, lepidium sativum, cannabis seeds, so many different kind of green methods which we have used. And I do believe more and more kind of nanomaterials must be utilized and developed in this particular category. Now, one important thing I really wanted to share at this platform is that why do research needs directions? Because the moment you do research with precise directions, they can lead to much more uh, better future, or you can say a bright future. So we have basically, although I'm not associated with this company, but I'm very proud of this one, the way it came up from the lab. Syria Therapeutics has a noble platform for microRNA delivery, and this Syria Therapeutics is a company which was held in Delaware, and it has office in Tucson, Arizona, Aurora, California, where lot of research into Syria Therapeutics is particularly going on. Not only on disease, we have they are working on very kind of disease like acute respiratory distress syndrome, acute lung injury, inflammatory bowel disease, and diabetic foot ulcer. So these are all the kind of different diseases on which they have been working, and this Syria molecule is working, and many of those stuffs are under preclinical trial. Industrial application is going on, and nearly in very soon or in future you would be seeing lot of drugs coming out in this particular future. So as an overhaul, your serum oxide material, it's a wonderful material with lot and lot of potential, and that is why the lab is particularly working in this particular category. And again I say that this particular material has a wonderful assortment. It can be in a very lucrative material in any kind of direction: antibacterial, antioxidant, neurotoxicity, anti-cancerous activity. So these are all different kind of material. These are different kind of research arena where your this particular serum oxide nanomaterial is particularly working. Thanks to my past and present affiliation, Center for Environment, IIT Guwahati, where I initiated my research, IIT Guwahati, Ohio State University, Amity University, University of Central Florida, where I did most of my work, especially the paper come from this lab, Nanoscience Technology, Advanced Material Processing Analysis Center, and definitely Sharda University, where currently I'm working, which is giving me a wonderful opportunity to initiate the research and everything into this particular serum oxide direction. And I would like to end my presentation with one quote, that there's nothing I believe more strongly than getting young people interested in science and engineering for a better tomorrow and for all humankind. The quote was given by Bill Nye, although I'm not a big fan of quotes and all those things, but I think this is the quote, is the need of time, the need of young people coming into science and engineering. And I'm very thankful to Dr. Abhishek, who is part of this Scholar Lab, whose vision is onto Scholar Lab taking the education, especially science and engineering, further to this particular category. Thank you, Scholar Lab. Thank you, MTTS chapter, this technical, for being such a great host and host me for this particular lecture. Thank you so much, guys. If there is any question from the audience, I would be really more than happy to take this further.
Thank you, sir.
Abhishek ji, over to you.
Thank you, sir. Thanks a lot. And we all are very much fantasized and like amazing. This whole presentation and the way you handle these things are very much amazing. Before our participants ask their queries to you, I have a question to discuss with you,
Yeah,
sir.
please, please.
Usually, I am from the deep learning background, so we do
Okay.
simulations and generate the validation and comparison with other papers
Uh-huh.
by the simulation and by the codes. So, what
Okay.
kind of experimental setup you opted and tools you opted to analyze the results and to gather the data and to observe the changes into the mouse and mice wounds, so that you can generate the statistical analysis or graphs, these kind of things. So what was the experimental setup and what was the things behind that analysis?
Yeah, mostly in that case, mostly the data setup and analysis is mostly biological. For example, you have to develop a wound onto a mice model. You cannot use any kind of software to develop that. So
Mhm.
you do punch biopsy, you create a wound onto the back head of this particular mouse. Now, in order to understand whether your material is working onto that category or not, so you have lab-based synthesis of serum oxide nanomaterial. You do some biochemical testing to make sure that your material is functionally active. The moment that material is applied onto those mice models and everything, and the kind of data you generate is mostly biological. But yes, the point is you do not have one mice, you have more than one. You have, let's suppose you have 10 mice into that category. So the moment you generate those 10 mice's data, you do some statistical analysis, and those statistical analysis are very preliminary, where you try to have an average, mean, standard deviation, t square, chi square, those kind of test analysis. So as of now, this material has, I understand your question, like what kind of software analysis basically you do. So as of now, these material haven't entered into those kind of software where we do lot of simulation based study. But I think that is one area where we can explore if we can have any kind of simulation based analysis on serum oxide nanomaterial, how it is scavenging oxide radicals, or how the radicals scenario is changing in a particular kind of solution. That area, I think, it still remains to be explored. I'm working in close association with certain bioinformaticians, where we are trying to understand if we replace a molecule in our body, for example, instead of a blood, if we can replace that particular molecule with any other kind of uh, serum oxide molecule. So, what kind of changes it will takes place. So, those kind of analysis are still, yes, it is pending so far.
Great, great. And what kind of expenses tentatively or the experimental setup a university, college, or in private research lab require to arrange these kind of testing? And any idea of expenses and kind of setup required?
Especially yes, when you do certain biological kind of experiment, there are a lot of expenses involved, because it's handling mice model and all those things are not easy. Lot of regulation is needed before you proceed. Lot of ethical committee approval is needed before you proceed. But yes, there are certain kind of expenses are needed when you do cell culture based study in a mammalian cell setting. You need an incubator, you need a specially isolated place, manpower, certain very highly or costly chemicals, those kind of requirement is needed. So I think that is something in India which not every college or every university has. But yes, that is something which is feasible, and the labs we have in Sharda University, we have an excellent DST, FIST facility. We basically call it as a FIST. And also in our department, we have couple of good facilities where we are able to do those kind of testing. I think in the next visit, whenever you have into Sharda University, I'll show you how these kind of materials we are preparing in our lab, and how the testings we are doing in our lab. That would be really wonderful for you to look into.
Sure, sure, definitely, for sure. Now, I would request our all participants if they have certain queries or anything to discuss about the research. And as Professor has said, their university is having a very much good laboratory to carry out these kind of experiments and research analysis. So I hope as even I don't have the permission from Professor, but still I understand that he is very much open to help out and collaborate with the researchers to work into this area ahead. So I would request our participant, if they have certain questions, they can go ahead with Professor Sushant right now. Or if later on also they have certain questions, then you can just write to me, or they can just browse the profile of Dr. Sushant on our platform and can directly into the touch with him about the discussion of their problems.
Yeah, science is an open platform, and I think we should be, irrespective of borders, irrespective of mode of communication, we should be open in science. And I think this is where new platforms like Scholar Collab, and I am associated with a couple of other different platforms, and I keep myself available over LinkedIn as well. Uh, it has to have an open border, and that this is where people will come connect, because we all are into learning phases. There are kids, there are schools, there are schools dropout students, there are college dropout students, there are university dropout students, there are the faculties, those who are into a very early stage of research lab development. We all need guidance, and even at this stage we also are looking for certain other kind of high level guidance where we look into our senior professors and other people around. So I think this is where the science should not have any kind of a boundary setup, any kind of limitation. It should be an open platform, and we all should be free in asking questions. We should not hesitate. Okay, fine, I'm talking to one a professor, what he would think, whether it is a question is a question. It is not neither big neither small. It doesn't have anything in it. If you have a question, you have a question. You need to come forward and ask it, because that's how we will solve the issues. Until unless we come forward, we ask question, we will not be able to solve that one. So I always tell my students when I do take academic classes over here, I always say, feel free to ask question, even if it's a silly one. It doesn't matter. It may be silly for you, or it may be silly for someone else, but it may be of a very big and high value for someone else who doesn't knowing anything. And I'm very proud to say that in my class, I have always seen those medicore student, a very average student, coming up and doing lucrative science. That is what I have always seen around. So I think this is where I always encourage the students to come forward and deliver whatever they can at the very best level.
Very well said, sir. Very well said. And I can see already there is a request to arrange another session with you.
Yeah, even I saw that when it would be really great to have those people, not only me, but there are other many people who would be love to come to this particular platform and deliver. And yes, definitely, by looking into the kind of audience we have and the kind of audience we reached into, although they were very less from all those places, but I think we can have a kind of a session where we can connect mostly those people who are willing to join. And definitely the session can go much longer, not only at this evening time, but it can go much longer as well if we have a very large number of participation coming out from those different reasons as well.
For sure, sir. For sure. All right. So I think we have no more questions from others. And if there would be anyone, then I'll try to connect them through email or through direct connection
Yeah,
with you.
definitely. Anytime, Dr. Abhishek, anytime you can reach to me.
Thank you, sir. Thanks a lot again. You have spared your time with us, and that's very much beneficial to hear about your knowledge and experience and exposure. And I must say, the presentation was very well, and especially
Thank you
these kind of
so much.
anatomies into life science are very much difficult to handle and to design even.
It's very definitely. But that's how the life is progressing. The way COVID came in and the way scenario changes, it was very difficult. So that's why I think this is where the intricacies of life sciences comes in, where they try to look into those kind of research, which is going to benefit for the future. The way diabetics cases are increasing, the way cancer cases are increasing, the way these post COVID complications are increasing. Like you can see there is change in environment, and especially right now, currently we are in NCR zone. You can see how the atmosphere of NCR is changing, and this all have negative effect onto the people. So the science also have to have very positive in order to overcome these challenges, and this is what as a scientist we all can do.
Very true, sir. Very well said. Thank you, sir. So, once again, I would like to share my gratitude with you.
And thanks to you, Dr. Abhishek, for having this platform. It's a wonderful platform. We do have lots of plan for this one. Let's take it further to the next level.
For sure. So great. Thank you, sir. Pleasures are all us. Thank you. And I would also request and again show my thankful to our all participants who has patiently listened to us. And definitely, I expect that they have gained something from your experience and presentation. Thank you, sir. And thank you, everyone, for your time. And once again, have a nice evening. Bye
Yeah.
bye.
Thank you, everyone.
Thank you. Bye
It
bye.
was lovely to see you all. Bye.